Hello and welcome to the third and final video on energy production, mitochondria and the tricarboxylic acid cycle. So in today's video we're going to understand the process of oxidative phosphorylation and how the mitochondria actually generate ATP. So oxidative phosphorylation is the process by which ATP is generated. In order to produce ATP, we need energy, and this energy comes from electrons which are transferred from NADH and FADH2, which are generated by the citric acid cycle. These are transferred to oxygen, which then goes on to produce water. This process occurs in the inner mitochondrial membrane. There are protein complexes in here, and in total there are 4 complexes, each of which passes on electrons, and each time they pass on electrons they release energy, and this is this energy which is then harnessed to produce ATP. So what does this look like? So here we have at the top we have the intermembrane space, and then we have our highly specialized inner mitochondrial membrane. So if we start from the left hand side, we can see our electron carrier NADH, and this is converted to NAD, releasing 2 electrons, which are then carried along through ubiquinone, passed on to complex 3. So the first one here is complex 1, this is complex 3, and we'll talk a little bit about complex 2 later on. Electron is then passed on to cytochrome C before coming down to complex 4. So complex 1 is the largest complex. This has over 40 polypeptide chains, and it's important to note that this has a iron or a ferrous ion at its center, which is important for the role. Next one is complex 3, or also known as cytochrome BC complex. This has 11 polypeptide chains, so it consists of 11 different proteins. This functions as a dimer, and again this complex 3 contains an iron. Finally we have complex 4, which contains a copper ion, and this is made of 13 polypeptide chains. So what happens as the electrons are passed on? So as the electrons are passed from complex 1 to complex 3 to complex 4, and remember this is all when we're considering NADH as an electron carrier, is the energy that's lost from these electrons as they drop down in the energy state is used to pump out protons through the complex. So these complexes, complex 1, complex 3 and complex 4, are transmembrane complexes. So the protons are pumped out from the matrix into the intermembrane space. As these protons are pumped out, we then create what's called a proton gradient, and it's this proton gradient that drives ATP synthesis. So the protons are positively charged and they will build up in the intermembrane space. And then there's a specialized protein called ATP synthase here on the right, which allows the protons to flow back through into the matrix. And as this is flowing down a gradient, it generates energy, and this energy is then used to create new ATP from ADP and inorganic phosphate. So the production of ATP or ATP synthesis is said to be coupled to electron transport. If ATP synthesis becomes uncoupled from the electron transport, and there are specialized proteins called uncoupling proteins, then this is how the mitochondria generate heat. So if ATP synthesis is uncoupled, then we generate heat, and it's this heat which enables us to stay warm as mammalians, But also it's important for animals that overwinter, they hibernate. So when they're not producing, they don't need lots of ATP. What they do is they use that energy to maintain their body temperature. Now I'm just going to show a short video which should give you a nice overview.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, two mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, two electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of one hydrogen ion for each electron. Next, the two electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. One hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from AD and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
And just another video.
Concentration gradients are a key component of the biological world. The potential energy from these gradients is often used to perform biological work. Here we will focus on hydrogen ion concentration gradients. Hydrogen ions are also known as protons. A gradient exists when there is a higher concentration of a molecule in one compartment compared to a neighboring compartment. This animation will demonstrate how the potential energy that results from a hydrogen ion gradient uses ADP and refined inorganic phosphate, also known as PI, to synthesize ATP. This process involves an enzyme complex called ATP synthase. Gradients and the potential energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins. Notice the large difference in the number of hydrogen ions on the two sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized. The process has now completed, and the result is an equal number of protons on each side of the inner membrane. Without a gradient, there is no more energy available to make ATP. In biological systems, however, a gradient is always maintained. The mitochondrial hydrogen ion gradient is generated as electrons pass through 3 membrane complexes. That process can be seen in the mitochondrial electron transport chain animation.
Okay, so we've now covered what happens with NADH. But as from the second video, you'll know that we also looked at another electron carrier called FADH. And you will also have noticed that we haven't mentioned complex 2. We've mentioned complex 1, 3 and 4. So what happened to complex 2? Well, complex 2 is not transmembrane and doesn't pump hydrogen ions. So we can see in the image below that obviously complex 2 sits between complex 1 and complex 3, but it's not transmembrane and therefore hydrogen ions cannot flow through it. Here is where FADH comes in. FADH will pass on its electrons to complex 2, and then from complex 2 these electrons are then passed to ubiquinone, which then passes on to complex 3. So in essence what we're doing is bypassing complex 1. And it is for this reason that FADH2 will yield less ATP per molecule, and that's because less protons are pumped out. It still gives rise to ATP, but you get less bang for your money. So here we have a multiple choice question, and again I'll read this out. So what type of gradient best describes the driving force of ATP synthesis in mitochondria? And you'll notice in here the key word is best describes. Is it a concentration gradient? Is it an electrochemical gradient? Is it an ionic gradient? Is it a potassium gradient? Or is it a proton gradient? I'll give you a second just to pause the video before I reveal the answer. And of course the answer is E, a proton. Now that's not to say a concentration gradient or an electrochemical gradient or an ionic gradient are incorrect, because that is one way of describing it. It's a proton gradient, and of course a proton is an ion. We have an electrochemical gradient because it's generating a sort of positive and negative charge across a membrane. And there is a concentration gradient because there's high concentration of protons outside compared to inside. But the best description for this particular one, and this is something that you must be aware of answering exam questions, is that we're asking for the best description, and the best description is the proton gradient. Here we have a typical short answer question. So, which organelle is responsible for generating the majority of the cell's energy? You get 1 mark, so that's a very simple, quick answer. State the location within this organelle where ATP synthesis takes place. And C, name 2 metal ions that are essential for this process. Of course, so the answers are to A, of course, is the mitochondria, is the organelle responsible for generating ATP and therefore the majority of the cell's energy. And the ATP synthesis takes place on the inner membrane. Remember, ATP synthase is the enzyme that's responsible for this, and this is found on the inner membrane. And of course, the 2 metal ions are iron and another one would be copper. Now in part B, I've also put up there on the cristae, and this again, if you'd written cristae, I would have also given a mark because of course the inner membrane folds, those convolutions, those invaginations that give it the large surface area are called the cristae, so you would also get a mark for that. So just to give a sort of overview of how these energy production pathways integrate, we have a nice sort of simple diagram here where at the top we have glycolysis. So from the breakdown of your food we get carbohydrates and glucose. Glucose is then broken down, releasing a small amount of energy in the form of ATP and NADH to produce pyruvate. Pyruvate can then be converted to acetyl-CoA inside the mitochondria. Acetyl-CoA will then enter the Krebs cycle or the citric acid cycle, which will generate again a small amount of ATP, carbon dioxide as a waste product, and of course the electron carriers NADH and FADH2. And it's the high energy electrons from these electron carriers which then pass through the electron transport chain here at the bottom, and this is what drives oxidative phosphorylation to produce ATP, using oxygen also to produce water. And finally, here's just another short video to give what I think is a very nice overview of mitochondria and ATP production.
Our body is made up of trillions of cells. They all require energy to function. This energy is created within our cells, in the mitochondria. Here, food is converted into chemical energy called ATP. ATP is released by the mitochondria so cells can use it. Mitochondria consist of 2 membranes, an outer membrane separating it from the cytosol, and an inner membrane surrounding the so-called matrix. The area between these membranes is called the intermembrane space. ATP is generated at the inner membrane of mitochondria by an efficient mechanism called oxidative phosphorylation, involving several membrane protein complexes. Nutrients provide high energy electrons in the form of NADH, which are used by the protein complexes to pump protons from the matrix to the intermembrane space. This continuous pumping creates a proton gradient, where the positively charged protons are attracted to the more negative matrix. When the protons re-enter the matrix Through the ATP synthase protein complex, they catalyze the production of ATP.
And that concludes the final video on ATP production. And if you'd like to read any more, again, I recommend chapter 17 of Straya. Thank you all for listening.